hydrogen nucleus with single proton having largest magnetic moment is widely studied by NMR spectroscopy. So the hydrogen nucleus is taken into consideration to study the NMR spectroscopy widely. For hydrogen nucleus, the nuclei can exist in two states. One is in ground state and other is in excited state. Again, we revise the basic principle of the NMR spectroscopy. When nucleus containing proton, so whatever the chemical compound having the orbitals surrounding in the structure centrally present at a nucleus in which protons are present. So when the nucleus containing proton absorb the energy, a signal is produced, and this signal is detected and amplified as a magnetic resonance spectrum. So up till now you don't come across with what is the actual meaning of the spectrum, how it is detected, how this instrumentation is attached with the computer facility and all this. So what the general mechanism, if this is the magnetic field present and in between this the sample is present and perpendicular to the magnetic field, from this direction if electromagnetic radiations are supplied, but initially in presence of magnetic field the proton containing compound show the alpha spin, and this alpha spin then go to the excited state to the beta spin when from vertical or from perpendicular region, when extra electromagnetic radiation is supplied, they change their motion. So due to change in the motion from alpha spin to beta spin, the proton containing whatever the chemical compound, proton containing whatever the biological compound is said to be resonating with the applied electromagnetic radiation which is supplied perpendicular to the applied magnetic field. So the original ground state go to the excited state, again excited state come back to the original ground state by emitting the extra energy or whatever the electromagnetic radiation. This is the basis of NMR spectroscopy. So whatever the chemical compound we are going to study by NMR spectroscopy, having whatever the number of protons present in the structure, due to presence of number of protons, they absorb the quantity of electromagnetic radiation which is required for resonating these protons from alpha spin to the beta spin. So this is the basis of NMR spectroscopy, or this is the basic principle of NMR spectroscopy. Now next point which is very important in this NMR spectroscopy is called chemical shift. In chemical shift, it is depending upon the surrounding environment present when the molecule will understudy, what type of another biomolecule or what type of different molecules which are present in the vicinity of the compound, whatever we are using for the study. Means if this is the X molecule, depending upon the vicinity, what type of different molecules they are present surrounding this X molecule, depending upon their molecular environment, depending upon their number of protons present in the vicinity of that molecule, this phenomenon takes place is called as chemical shift. So there may be change in the spectrum obtained due to the electronegative atoms present surrounding the molecule, or there are different mechanisms depending upon what type of molecule present in the vicinity of that chemical compound of which we are going to study the spectra. So the chemical shift in this, the nuclei of an element give distinct spectral lines depending upon their surrounding nuclear environment. Means whatever the molecule which we are going to study by the NMR spectra, surrounding to it what type of different molecules are present, or surrounding to it what type of different biomolecules or chemicals are present, which are responsible for either attracting the protons from the nucleus or which are responsible for shielding or masking the protons from the structure of which we are going to determine the spectra. So the spectral line behavior or what type of spectra obtained in the result may vary depending upon the surrounding molecular environment present, which is present near this central molecule of which we are going to study the NMR spectra.